Okay, good morning, everyone. We're going to go ahead and get started. Hopefully, everyone got into the meeting okay. I see there's still quite a few people joining. My name is Erica Olson. I am the Farmer Network and Communications Coordinator at Discovery Farms, and I'd like to welcome you all to the Northeast Regional Tile webinar. This meeting was originally going to be an in-person meeting held at the Manitowoc County Extension Office, but due to the current environment we are in, we switched it to a webinar. So hopefully we can still get the same impact and good presentations. And I think it might have actually been able to increase our reach and get more participants as we could get more people statewide, and location wasn't a limiting factor. So thanks for joining. I am recording this webinar and I will post it on the Discovery Farms website later this week. If you are a crop consultant hoping to earn CEUs from this meeting, please type your name into the chat box and we will get those recorded. And with that, the Discovery Farms program, for if you're not as familiar with us, we are a on-farm research and outreach program that's farmer-led. We are part of the Division of Extension through UW-Madison. And we do research across the state focusing on water quality. Today's meeting is going to be focusing in on one of our projects. It's a tile project that began collecting data at the edge of field for tile sites back in 2018. Aaron Wonderlin and Eric will dive into results from that project regionally. It's taking place on the eastern side of Wisconsin. And so today's meeting will also have Matt Ruark, who will talk a little bit about some soil health, a soil health piece of the project that we've intertwined with the tile monitoring project. And we'll also have Steve Huffman, who's going to talk a little bit about tile. And so with that, I see there's still some people joining, so we'll get those people admitted as fast as we can. And our first speaker is going to be Matt Ruark. He's part of the Department of Soil Science at UW-Madison, and he's also the faculty advisor for our Discovery Farms program. So Matt, you can go ahead and share your screen.
All right. Thank you, Erica. So, so my name is Matt Ruark. I'm in the Department of Soil Science. And so I'd like to also acknowledge my co-author on this talk, Chelsea Zegler, who has put a lot of stuff together. So you've probably seen a lot of soil health stuff through the year. So Chelsea presented this similar talk at the Cover Crops Conference. Steve Coleman from Ohio State presented on some soil health stuff at our Wisconsin Agribusiness Classic, and then I presented back in the fall related to. Oh, sorry, Erica, did you mean to pop up the question? Anyway, so we thought we'd start with this polling question. And so for those that haven't done this before, you can just click right on the screen there. And so just to get kind of some people thinking about it, think about what soil health building practices do you use on your farm, or I came up with at least a quarter, 25% of the practices that your clients use. So here we got, click all that apply. You can use multiple choice. You can click on as many as you want on this, but perennial crops in rotation, manure application, cover cropping, reduced tillage. We include seasonal grazing. That's not that may be a little more common out west, but and also properly managed pH and nutrients. So you can see a lot of getting a lot of responses. Looks like actually reduced tillage and cover cropping are leading the way right now. So, and this is important to, and the reason why I'm asking, and I am certainly can keep filling out the survey here, but there's a lot of different practices that go into soil health, right? Or a lot that are beneficial to soil health. And a lot of them we may be using already. And maybe there's an aspect of in Wisconsin, because of our dairy production systems that have perennial crops, that where we can have some reduced tillage in areas where we are using manure and we have some opportunities to use cover crops. Maybe, where we can be a little ahead of the game in our ability, as opposed to other states that are maybe a little more focused on corn and soybean. So I'm gonna go ahead and give you one more second to click on it. I'm gonna end that poll. So if people are interested in what the survey results are, Erica, can you see those results? I have no idea if this is working or not. So we have, looks like reduced tillage. So 73% of the respondents have reduced tillage, 71 have cover cropping, 71 have manure application. So a lot of those. So that's good. So as we move on, here we're gonna end that poll. All right. So as we talk about soil health, and this is some slides I took from Steve Coleman at Ohio State. We talk about soil health as a single thing, and we talk about soil health as a single thing, and it's a whole series of things. With is it a philosophy, right? Is it an endpoint or a journey? Are we trying to get to soil health? Is it a series of farm practices and management? Is soil health something you do and actively work on? Is soil health a series of measurements in the field or lab? Or is it feel-good marketing fluff, right? And so, we just went through those farm management practices, and those are aspects of soil health. And what I'm going to focus mostly on today are these ideas of the measurements of the field, these lab measurements of soil health. So as we go on, I'm happy to take questions throughout. If anyone has any questions, feel free to type them in that chat box, and hopefully Erica will chime in when needed. So what does this healthy soil consist of, right? So everyone's got their own ideas too about what soil health is. Is it sufficient nutrients? It has good tilth. It's easily workable, easy to plant. It's got sufficient rooting depth. It's got good drainage and all these physical properties of it. In some biological aspects, it has few pathogens, right? It's got beneficial soil biota, so we want more good stuff and less bad stuff. Low weed pressure and no harmful chemical or toxins to the crop. And then lastly, it's resilience to degradation. So as we get into parts of the season where they're more prone to extreme weather events, how does that soil hold up? So soil health is multivariate by nature. And so it means that we are going to want to measure a lot of things about the soil. But it's not something we can sum up. It's not something we can just keep add up values, right? So we need to think about the chemical, physical, biological. I'm sure everyone's seen this, and if you've seen my talk before, I like to give these little snippets around it. The chemical aspect, these are well studied, they're easy to measure, pH, soil nutrient levels, and then we know what to do with the information, right? We have recommendations built around pH, pK, that sort of thing. The physical properties are also well understood, right? And they're often things you see. You can see surface sealing. You can certainly, I don't know if you can see compaction, but you can dig and you can put a shovel in the soil and understand how compacted your soil is. But they're often hard to measure in terms of the porosity of soil, for example, is very cumbersome to measure. And lastly, we have the biological properties, right? So these are not well understood. They range in ease of measurement. The ones that I'm going to talk about today are pretty easy to measure, but there's a lot of biological measurements that are that are more difficult. And then the idea of what should we measure? What should we be looking for? So if we want to manage it, we must be able to measure it. So we can think of all these things, this whole list of chemical, physical, biological indicators. What do we measure, right? So what I did here is I've pared it down to, we're going to focus in on the biological measurements. So these are the ones that we think have some value, something that we can take a soil test or soil sample, send it to a lab and get results quickly. So I like to show this. There's three big players in the soil health game: the USDA NRCS, the Soil Health Institute, and and Cornell's Soil Health Lab. And so what I have here is that I have different biological measures on the left here, and then I marked which ones are recommended by each group. And I want to delve in more specifically on these four: this mineralizable carbon, mineralizable nitrogen, the soil protein, and the active carbon. Let me go back one. So the organic matter, organic carbon are organic matter is a pretty standard measurement, and organic carbon is just a more specific measure of the carbon content of the organic matter. And then the ones on the bottom here, the PLFA, the enzymes, are a little more cumbersome to measure. So these four in the middle are ones that most of the groups recommend and would fit easily into a soil test lab. So what are these measurements? So we have incubation measurements, and we've got soil extraction measurements. So the incubation measurements are the ones where we're going to let the microbes do the work. So this will be soil respiration, CO2 burst test, mineralizable carbon, a lot of different names for it. But it's basically just over a 24-hour period, how much CO2, how much carbon dioxide is produced by microorganisms after 24 hours. So it's a simple test. You take the soil, you dry it, you dry it down, you put it in this container, you wet it back up, you seal it, and then after 24 hours, you measure the CO2. And we're going to use that as a proxy for microbial activity. The other one's potentially mineralizable nitrogen. So same thing, we're going to measure the conversion of organic nitrogen into ammonium under anaerobic conditions. And we're going to use that as a proxy for the nitrogen supply from the soil. The PMN's a little bit longer though. That's a 7-day incubation. So there is some time involved. It's not is 24 hours a rapid test? That one's might be on the fringe. The mineralizable nitrogen is a longer test, unfortunately. The two then that are what we'd like to do then is, instead of letting the microbes do the work, is there's something we can try to capture about the soil with a chemical extraction that gives us the same meaning. So chemical extractions, obviously, you can do in bulk, and if we're just doing colorimetric analysis on it, it's something that we can do pretty quickly. So the first one is this POX-C, right? Permanganate oxidizable carbon. Often you may hear it called reactive carbon. You may hear it called active carbon. It's basically just the chemical extraction for a labile carbon pool, so a readily accessible carbon pool. And then that is a proxy for a microbial food source. So then the other one is similar to that. Then there's an organic nitrogen pool that we can also do a chemical extraction of. This is the autoclaved citric extractable protein. So soil protein in the soil, it would be a big pool of organic nitrogen and be one that is likely to become plant available nitrogen during the growing season. So it would be an extraction of potentially available organic N. These methods and the chemical methods are better suited for commercial soil testing labs. So I do want to mention the Haney test. I know I end up getting a lot of questions to that at the end, and I have not I haven't done work with the Haney test directly. So when people talk about the Haney test, there's two different components of it. So technically there's three. It's a series of things. You have the mineralizable carbon as a component of it, that 24-hour burst test. So that is part of it, and we are measuring that as part of these studies. They also do this water extractable organic carbon and organic nitrogen pool. So it's just a water extractable pool. Then they do a weak acid extractable thing for carbon and other nutrients. So then you can do two things with it. They do use this to make some fertilizer, or some labs are using it to make fertilizer recommendations. I don't have much insight on that, on how that works. But they do use these first two values, the mineralizable carbon and the water extractable, to develop this soil health calculation. So they take your min C divided by the ratio of the organic C to N, and then you add the water extractable C divided by 100, water extractable N divided by 10. You add those together and you get a soil health value. And then, so I've been using the Midwest Labs website for some sort of interpretation on it, and what they indicated here is, what they're promoting is the values. The goal is to see an increase in that number over time. So You're not going to see a lot of stuff on the overall Haney test, but components of it, specifically the mineralizable carbon, we'll have some more discussion about. All right, so Erica, can you pop up the next set of polling questions? Got some more questions for the group here. Where do I have to do that? Maybe I'll do it.
Can you see it?
I see question 1.
Do you
I don't see
test for
question 2.
soil health?
We're not seeing it out here.
There we go. Maybe I can do it, Eric, I think. All right, how about that? You seen that? Do you currently test for soil
I'm not
health?
seeing it, Matt.
You're not seeing it? Shoot, I wonder if Erica needs to.
No, we can't.
Looks like it's live.
Mhm.
I'm not.
I'll turn that off. I wonder if okay. Well, we can give one more second and see if we can figure this out. Hey, there we go. I'm seeing it now.
No.
Okay. So we even got a response. All right, yeah, so let's ask. All right, so we just went through some of these soil health tests, and there's some labs out there that will measure soil health. For the people on this webinar, do you currently test for soil health? Just simple yes or no question. So I'm seeing these results live, and we're getting a good chunk here. So right now, the vast majority are no. I'm going to be a little quick with the gun on this one. I'm going to end it now. And so you can see that about 80% aren't, but a good 20% are. So 10 people on this webinar are testing for soil health. So it's not out of the realm, right? So these are things people are testing for. It's not just one crazy person out somewhere doing it. It's a thing that people are interested in. That's interesting. And then
next.
Yeah, let's go to the next one. Then my next question for the group.
Can you see that?
Yep. If you would test for soil health, this one you can pick just one. If you would test for soil health, what would you use the results for? So we have you're not sure yet, but you're sure you'll figure it out. You have some ideas in the future. To track the changes over time, so it's a long-term metric. To understand the yield potential or the value of your soil, is something connected to economics. To guide soil management decisions, for example, if you want to implement some soil health building practices. To guide nitrogen management decisions, that could be other nutrients as well, but the big one that's typically we try to connect to is nitrogen. Or you're going to collect it, but you wouldn't use the results for anything. And I think the reason, a lot of the reason why I'm asking this, in the poll here, we can take a look at the results. So, number one was to track changes in soil health over time. About 41% of the respondents said that. The second one is to guide some management decisions, about 31%. So those being, and then third place, to understand the yield potential or value of my soil. So I think that right now we're in a spot where we can use these soil health. The value for right now, I think, is in that track changes over time, right? It's something to monitor. Do we have good benchmarks to know what these critical thresholds are, or what average values are? We're slowly getting there. So what we'd like to do, in my line of work, is really that number 3 question, to understand that yield potential of the soil. What's the connection between soil health and yield? And then lastly, what is that? Can we use it to guide any nitrogen management practices? All right. So the next 3 things I'm going to talk about here, then, are these indicators sensitive to management? So are they good soil health metrics anyway? These 4 that I'm going to talk about. Are they connected to yield in any way? And then are they connected to nitrogen management in any way? Or I should say, are they connected to are they connected to optimal nitrogen in some way? Can we use them to guide fertilizer recommendations? So what has UW data shown us so far? So the idea of being sensitive to management, the first thing we're going to use is some of our long-term experiments. And so, and I will go through this a little quickly, and I guess I apologize up front on that. We've got these trials where we have continuous corn, soybean, and corn, soybean, wheat rotations that include these annual and then perennial systems of corn alfalfa or rotational grazing. And each of those have an aspect of diversity within each system. So we collected soil samples in the upper 6 inches. This is at our Arlington Research Station. This has mineralizable nitrogen, mineralizable carbon. So these annual cropping systems in the orange and the perennial cropping systems in the gray or in the green here. And you can see for the most part, things kind of separate out. So if you have alfalfa in rotation, you have some level of perenniality. Deep-rooted perennial crops, you tend to have slightly higher values compared to annual cropping systems. And in the mineralizable components, in the incubation components, the pastures really stand out as different than everything else. But actually for the POC, the chemical extraction, it wasn't that different. So some systems or some measurements are showing big differences among cropping systems and others don't. Well, interestingly, we found that sometimes it depended on when we sampled. So an ideal sampling time would be the spring. So you have everything come out of the spring, and that would be a pretty standard time to evaluate your soil biology. And so here's when we only sampled in the spring, and this is from our potentially mineralizable nitrogen. You can see that the corn alfalfa rotation had the highest values and wasn't different from the pasture. So the pasture and the corn alfalfa looked very similar. In addition, the continuous corn looked much greater, had greater values than the corn soybean rotation. But when we went back and we sampled in July, one is that this was when we typically saw peak values for most of our systems. But we see now that the continuous corn and corn soybean weren't that different. And the pasture really became much greater than the corn alfalfa rotation. So if we would have just gone out and sampled one time, and we would have come to completely different conclusions about these cropping systems and the value of these soil health metrics. So then, what does this mean for the Midwest? Where our Arlington Station is pretty, it has the similar soils throughout the Midwest. So isn't a big enough change in diversity to really influence soil health. So going beyond corn, soybean. And then I just if you want it, the biggest impact is we need to try to make our annual systems look like perennial systems. So that's going to mean reduction in tillage. That's going to mean use of cover crops. And that's also, or animal agriculture, right? So getting animals in the landscape, if not physically, at least their manure. The timing of the sample is important. We don't know yet. We've been sampling, in most of our studies in the spring just for a standard sampling time, but it's not entirely clear when the ideal time to collect these are. Typically speaking, we see greater values in the middle of the growing season. So once everything warms up, the soil biological activity gets a lot greater, and then the values are a lot greater. So what about outside our Arlington Ag Research Station? So first of all, we're going to use an unstructured sampling design, and we just worked with as many farmers that would let us on their fields to sample their fields, right? So we worked across Minnesota and Wisconsin. You can see these clusters of area where we sampled farms. In this, we typically sampled in mid-April before planting, and we always sampled to 6 inches. So we've got a data set of 218 fields. Not only that, we also got a lot of information about the soil properties on each field. So the texture, sand, silt, clay content, the drainage class, how well drained the soil was. And then we also asked the farmers about management. So what was their tillage operation? What was their crop rotation? What were their manure inputs? Did they use cover crops? Other things, was the field tile drain? Was it irrigated? And those sort of things. So what we can do with this is that we can use something called regression tree analysis to figure out what is the most important factor in play that's influencing these biological measurements. So first we're going to start with just your typical soil organic matter measurement, right? So what we determined then based on and from all this work, what was the main factor that drove the organic matter level? So what we have on the screen right now, our average value across all our sites had an organic matter of 2.9. The number one factor that influenced organic matter was the clay content. So heavier clayed soils had much higher values than our lower clay soils. And you can see that when we had greater than 26%, we had an average organic matter of 5.2. So we had this data, we had a series with a bunch of data within there that had really high clay contents that had much higher organic matter. Okay, so that's driven by inherent soil properties. Well, then we can look at, we can split this data set and what's the next most important factor? And it was the crop rotation. So that would be if the fields were typically under continuous corn or included alfalfa. And so these two paired up because they're generally going to be the ones that have more carbon inputs into the soil. So continuous corn has a lot more carbon returned to the soil through corn stover and those sort of things compared to soybean and wheat, even though adding those in helps diversify the rotation. And then of course alfalfa, deeper rooting, and you have reduced tillage and stuff like that. So if it was those cropping systems, we had much higher values. And then lastly, we had one more split on this side. If it was in continuous corn or alfalfa rotations, if the field was tile drained, it had higher values. It was just a smaller data set here, smaller percent. But if the field was tile drained, it had greater values. So the trouble with something like that within this analysis is the idea that if there was tile drainage, is that causal? So the fact that the field was tile drained, did it make organic matter go up? I suppose you could, if yields went up because it drained, perhaps. Or is it because if a field needed to be tile drained, it was typically pretty wet, right? It probably had pretty high organic matter to start. And then we also have another, texture was also another player on that side. So I don't want to go through all of these, but I'm going to give two more examples for our mineralizable carbon. So this is our incubation measurement. Our first measure, so while organic matter was typically driven by the clay content, mineralizable carbon, which would be a soil health metric, the primary factor was a management factor. And now it came out that continuous corn or included alfalfa having higher values than those that didn't. So tying in really this idea of carbon input into the system to build biological activity. Although interestingly, then the second split over here, there weren't any other splits other than this tile drainage split. So it could also just be a couple tile drain fields that had really high values, but that was it. So it was really on our data set, the only real important factor for mineralizable carbon was crop rotation. So this kind of builds into a couple different things. So the first is, do we have enough data points, right? We only have 216 data points. Is that going to be enough? If we had more data, I think we would be able to build larger data sets. So when we talk about this idea of big data in agriculture, certainly it's more than 216 data points. We're using some of these big data analytical techniques. And what feels like a lot of data, it took us many years to collect this data and a lot of analysis, but in the end, we'd love to have data sets in the thousands to really pull out more factors. I'll kick out one more thing from POC, and this is messy. This is kind of a little bit fresher data we worked up. The number one factor for POC was pH. So interestingly, which can be a management, could be a result of management, or it could be an effect of the inherent soil properties, but typically pHs of greater than 7 had higher POC values. Then as we go down, the clay content and crop rotation were also players in this one. But so if we think about pH and clay content or region of the state as the main factors, POC tended to be a little more influenced by the inherent soil properties compared to management practices. Let's see here. Oh, okay. So why was there no effect of other soil health building practices, right? So why wasn't, we talked about manure, we talked about cover crops, we talked about tillage. That didn't come out in this. So what's going on? Well, it really relates to the nature of our data set. manure is applied on the vast majority of the fields, and then the way manure is applied and the source and the rate and the frequency was pretty variable. So there's so many factors that go into that it probably just contributed to the variation. So we didn't have enough, really, I guess we didn't have enough non-manured fields, if that makes sense. Also, cover crops didn't come out. It was grown on 31% of the fields, though, interestingly, but about 3/4 of those only started cover cropping in the last 2 years. So if we talk about seeing the benefits of cover crops for soil health, that's a long-term game as well, right? So we just don't have the long-term cover cropping aspects in this data set. Tillage, now tillage was interesting too, because it was very regional, right? So about 2/3 of our no-till fields occurred in the driftless region. So in areas where people are using tillage to save, to prevent erosion on their soils, where they don't have, as deep of an A horizon. So is that because of the tillage, or are people using tillage on certain types of soils? And those types of things we couldn't parse out either. So that's not to say these things don't build soil health. There's a lot of other studies out there that show the long-term benefits of these things, but in our data set, it was confounded with other factors. So were these, are biological indicators of soil health useful in any way? So what we've also been able to do is try to connect these soil health metrics to yield and to optimal nitrogen. And so warning here is that it's a lot of complicated statistics that don't translate well for extension talks, where we're using, we're basically building models out of data. We're using structural equation modeling or stuff like that. And it's hard to interpret. So we can interpret these things as sort of, if you trust in the statistics, here's what we found. So we used the Discovery Farms data set to understand the relationships among all the soil variables we measured and the nitrogen applied on crop yield, on corn yield. So in the model that we put together, soil organic matter was an important variable that drove yield. So the two most important factors in this data set were nitrogen applied and yield. Now, in our data set, the soil health variables weren't able to add much more to the model. So the organic matter contribute a lot to the model, but the soil health variables didn't add much more. So we ended up only using organic matter in the model. This could be the idea. It also reflects the fact that we need bigger data sets. But on the graph here, we have the yield. So in metric units, of course. Sorry, we submitted this for publication, and this is organic matter. And then we had 3 scenarios in our model where we have no manure, no nitrogen applied at all. Manure applied as a combination of fertilizer and manure, and a combination, or entirely as nitrogen, as inorganic nitrogen. And you can see when we're not applying any nitrogen to the soil, the organic matter has a great contribution to yield, right? It increases it pretty rapidly as you go between 1% to 2% organic matter. Great benefits, and then that benefits slowly still increase, but taper off as you get larger. So to be fair, we did extend this out to 6.5. We had only a handful of data points really above 5%, 4.5% to 5% organic matter. So the data on this side of the graph is a little more sparse, but you can see still there's a positive relationship of building. Even when we're adding nitrogen to the crop, there's still a benefit on top of the organic matter as well. So increasing organic matter can help improve yields at any given nitrogen rate. So another thing we did is we worked across the Midwest with a group of researchers, and we put our data sets together, but not our Discovery Farms data sets. These are our nitrogen rate data sets. And this to address this thing: does soil health lead to reduced fertilizer requirements? And what did we find? So the trouble here is that we have this idea about soil health and its direct contribution to yield. Is it having is it something about there being more epoxy in the soil that's stimulating the plant? Is it a direct effect, or is it an indirect effect? By more biological activity is probably going to mean it has, is it influencing available nutrients and stuff like that? So is it having a direct effect on the plant, or an indirect effect on the plant through other processes? And I'll save you all the boring statistics, but here's our paper in Scientific Reports titled Improved Soil Biological Health Increases Corn Grain Yield in Nitrogen Fertilized Systems Across the Corn Belt. So here's our main things, is that it was the POC and the ACE protein were these soil health metrics that actually that we found to be important. And so we determined they can be used in conjunction with inorganic nitrogen, like a pre-sidedress or pre-plant nitrate test, to predict if a site will be responsive to nitrogen. So this was the idea of responsiveness. Unfortunately, we couldn't dial it in with this data set to the optimal nitrogen rate. The other aspect of this is the mineralizable carbon that the incubation one was not a valuable factor in our model. So this data set and the way we analyze it would indicate that the soil extractions actually were more useful than the incubation. The second one is that any given nitrogen fertilizer rate yield, increased soil health will increase yield. And so this is a little bit similar to that last data set we showed, where as long as you're fertilizing, at any one rate, having more organic matter at that rate will lead to more yield. So organic matter and soil health. So it does show that building organic matter and building soil health is important and can be beneficial. Now, overall, the biggest benefit was fertilization, right? And so that's the biggest effect on yield was fertilization. And there was this loop back in the data where it was really the fields, as long as we have good, applications of nitrogen to corn, can improve soil biological health in the Midwest. So maintaining good yields with optimal nitrogen rates has a positive feedback on soil health. So, which really comes from the idea of contributing greater biomass, more corn biomass going back in. So all of these things start to tie back in together. And then lastly, we've got one more paper that came out of a different group of researchers in the Midwest. We're evaluating that Haney soil health tool. And what we have here is among all these nitrogen rate studies, so they looked at that soil health the overall soil health calculation, or they just looked at that Solvita burst test, which is that incubation, that 24-hour incubation, and graphed it against that optimal nitrogen rate, the economic optimal nitrogen rate for that site. So these are all the sites in the Midwest that were conducted as part of the studies, and they found this relationship where the greater the soil health calculation, or the greater the CO2 burst test was, the less, the optimal nitrogen rate was less. So fields that have greater microbial activity required less nitrogen to optimize yield. The R-squareds are right in kind of that middle zone where we don't know what to do with, right? R-squared of 0.55, it's not overly predictive, but it certainly is adding something to our understanding about economic optimal end yield. So this data set would indicate the incubation would be beneficial. I don't believe they measured any of the extractable components though. So in the end here, we've got this great data set where we can benchmark some of these values in the Midwest or in Wisconsin, and we can figure out if they're responsive. But we've got these three papers that kind of conflict with each other. So the Wisconsin-based data shows soil organic matter is the best predictor over these other biological components. Others don't make the cut. But that could just be we don't have a big enough data set yet. In the Midwest, across the Midwest, we determined that POC and ACE protein, those chemical extractions, were useful and connected to yield in some way. This other Midwest data would suggest that the mineralizable carbon was a useful addition to soil testing and to guide nitrogen applications. So, are you confused? But I'm certainly confused by this, but I think that that's okay. I think that the takeaway then is that we have some exciting things that are happening and data sets are being built, and the early indicators are all of these things are going to be beneficial and be connected to yield and used to guide nitrogen applications. And so as we start to build these data sets even more, conduct more on-farm research, these things will be built and gives us at least a positive sense of what this thing is, what we're doing is beneficial. And if I can just have one more quick minute, I'll give a recap of this. So the value of these biological measurements as we break it down: Epoxy is sensitive to management, but so far we can only show it's beneficial, sensitive to crop rotation. It may require interpretation based on soil pH. There is some connection with corn yield, and potentially useful in predicting if a site is responsive to nitrogen. How responsive to nitrogen and how much nitrogen apply, we don't know. But if it's responsive, is a good. We can at least go to that level. It's generally has been untested as a predictor of nitrogen need. The ACE protein, now that's the chemical extraction for that organic nitrogen pool, right? Relatively untested in Wisconsin. I would say this is a new addition to my lab in terms of the things we're measuring. So I like it as a complement to the POXC measure, and we're getting all of the studies moving forward are going to include the ACE protein analysis. We do have some indication that's connected with corn yield and a lot of the same things with POXC, potentially useful in predicting response, but generally untested as a predictor of end need. So the mineralizable carbon, highly sensitive to management. Again, so far, really only crop rotation, but may require interpretation based on texture or drainage class. Limited connection with corn yield, but maybe higher, in contrast to the extractions, may be more beneficial in predicting optimal nitrogen need. And then potentially mineralizable N, highly sensitive to management, may require, so same things with the MinC, but untested really with corn yield. And really it's the last one here, it doesn't seem to be a popular test outside of the work I'm doing. We didn't include it in any of these other studies. My theory is in the stuff we've studied so far, it has been responsive in a very sensitive indicator to some management practices, but really maybe it's more useful in terms of manured soils. So that's why it's being picked up, because we have these other mineralizable nitrogen pools that we're capturing. So we'll continue to work on that. So lastly, if I can leave you with anything, and I did go over time a little bit, but soil health is still a young emerging field that needs refinement, absolutely. It needs soil specific type recommendations, and the value of the measurements are over time it's not a linear path to success. It's gonna be a lot of confusing stuff that's gonna happen, but I think we're at least pointed in the right direction. And so I guess as you are building your own data sets of soil health on your fields, these measurements will be valuable to track over time. As we continue to move forward, which ones will come out as more valuable and less valuable, we're not sure yet. So with that, if anyone does have any questions, I'm happy to take them. I know I went way long on that, but
We have one question in the chat, Matt. What was the average number of years soil health practices were used or implemented within the multi-state data set?
Okay, so for so for cover crops, right, it wasn't that long, so that tended to be for tillage. We did classify things as a no-till site, so it had to have been no-till for we used 8 years as our number. Was it no-till for 8 years or more? If it was no-till for 8 years or more, we call that no-till. And then for manure, fields were generally either manured or not, and then it was applied every other year or not. So there's so much nuance in that, and as we built our data set, we sometimes had to lump stuff into categories. As we're continuing to build out this data set, we can have more subcategories, like was it liquid dairy manure or was it solid, or was it solid manure, right? Those sort of things we don't have enough data to parse out yet, but we're looking to do that. So that's a good question. A lot of this will be data set focused. So building data sets is a key component of the future of this research.
Great, thanks, Matt. I'm not seeing any other questions in the chat box at this time. So I think we'll go ahead and move on to our next speaker. Next, we have Steve Huffman. He is joining us from In-Depth Agronomy, and he will be talking about tile systems mapping.
All right. Looks like I need to share my screen, and it's telling me I can't right
you should
now.
be able to now if you try again.
All right. Can you see my presentation on the screen?
Not yet. Did you click the green arrow in the middle bottom?
Yeah, share screen. Okay, now I've got that. All right, we should be good to go. Can you see that?
Yep.
Okay. It should.
Yeah, there we go.
There we go. Okay, so I've been asked to share some information of a tile mapping system that we've used in our company for the past, probably 10 years. And so why develop a tile drainage system map? We all know that the tile system presents a hazard to surface water, and nutrient management plans require a setback or an immediate incorporation zone around the inlets and vents. We know that outlets require monitoring during and after manure application. And we also know there's a lot of, at least in eastern Wisconsin, a lot of clay and concrete section tile systems that were installed 40 plus years ago, and those systems have been showing their age for quite a while now. Tile blowouts and suck holes are a hazard to surface water quality during and after manure application. With a good tile map, it's relatively easy and quick to check for tile blowouts. Having a map of the existing tile drainage system is also very helpful when planning an upgrade. And we all know there's been a lot of interest in grid tiling, but with the economics now, sometimes farmers will maybe look and ask themselves, well, can I just add an extension to the current system? And having a good map is very helpful when planning that out. And lastly, if you've ever stumbled through tall grass for the length of a ditch looking for an unmarked tile outlet, you will recognize the value of a good tile system map. And like the previous comment, they're hard to find. You know, tall dead grass or tall green grass, and it's not easy to find them, but a good map can really help you. So the system that we use is, I like to call the jigsaw puzzle method of tile system mapping. And what we need to do this is a moderately robust GIS program on your desktop computer. And there's several that would work. We've used DeLorme XMAP, QGIS, and Esri ArcGIS. You're also going to need access to multiple layers of georeference imagery. One of our go-tos is NAPE, the National Agricultural Imagery Program. And we also need a mobile GIS app on a tablet device or on a smartphone. And then you also need to think of this as kind of a long view systems approach, that you're gradually going to build this tile system map. It's not going to happen overnight. And my next comment makes us think that, well, maybe it can happen quickly, because there is machine learning programs that I believe are going to quickly develop maps of apparent tile lines by using multiple high-resolution satellite or aerial imageries. But what we've got to realize is that word apparent. Ground truthing is still going to be very important, no matter what method we use to come up with these maps. And this just references that previous comment, doing some searching, that this group here did some research and found that they could have a really good correlation just looking with Google Earth images and machine learning to draw out tile maps for fields. So we're all familiar with this. Basically, the more images, the better. They need to be georeferenced, brought into a GIS program, and the method we use right now is just sit down and look at what looks like it could be a tile line and draw it in manually, and then put up the next image. And that's why I called it the jigsaw puzzle method. It's slow, but it has utility. So same area, different year, and now we're all familiar with the kind of images we get from Bing Maps or Google Earth. And one of the things to point out is, in the center there, this field with bare soil, the tile lines show up quite well, but on the field next with vegetation, we're not seeing anything. And that's one of the reasons for, multiple years, but it's also one of the challenges as we get maybe a field that we know has tile, but it's in permanent vegetative cover, or as we get more and more cover crops, our window with bare soil is going to be less, which is a good thing, but it presents some more challenges. So we're going to have to maybe rely more on historical images as we get more ground cover. And also, yield maps are a good clue to where there's possibilities of tile lines. This yield map is one that we processed for our client, and we already had tile maps for that client. And I was looking at the yield map and thinking, boy, that looks like tile lines to me, because it was a wet year. And so up in the upper left-hand corner, you can see a green line that extends through a yellow section that would indicate to me there's probably a tile line through there. So it's an opportunity to look closer at imagery and see if we can't find that tile line. So once you've got a map drawn out, it's just apparent lines. It's what we think the tile system looks like. And that map needs to be refined. So what we found to be very useful is to give a paper copy of those maps to the farmer or landowner and ask them to cross off the lines that are not associated with drainage, and ask them to also draw in the drainage lines they know to exist. At the same time, ask them to also draw in the known location of inlets, vents, and outlets. And that step right there will greatly increase the value of your tile line map. It'll save you a lot of time and point out things that you might not find otherwise. Then the next thing is ground truthing. That's a very important step. No matter if you did it in the jigsaw puzzle method, or if there's going to be an app someday that spits out tile line maps for you, it's still going to be important to ground truth that map. And to make that easy, we need to load that onto a tablet computer with the GIS app. And there's a lot of apps out there that would work for this purpose. So this is a picture of the current tool that we use in our company. We use Android tablets and an app called Map It Fast. What you see there, the lines are apparent tile lines that we mapped out in that system. We put them into this app along with the field borders, and we can zoom in the field, drive those lines to the ditch or waterway, whatever it is, and verify, is this real? It's at the same time with this app, we can take georeference photos. We can add points if we found a tile blowout or wanted to add a line that we see as a dry line on the soil. We could do all that with this app, and the data would go up into the cloud and get added into the system. So really the best time to ground truth your tile map is right now, this time of the year. This initial spring green up is a great time to ground truth. It's a great time to look for tile blowouts. What we do, like I said, is drive if this is the first time checking your map on the field, is just drive the field perimeter, look for vents, look for inlets. A lot of times these plastic vents will be, deteriorated, busted down, and hard to see once the vegetation grows. Here's a picture of an old outlet that's still functioning, but try to find that without a map would be almost impossible. And so I mentioned the georeference photos. That has added a lot of utility. As you go around and document things, take a photo that can be put on a map and kind of refresh your memory on what is where and what needs fixing and those kinds of things. So the system is really meant to be used year-round. We use it as we scout. So here, on knee-high corn, you looked off in the distance there, you can see a crop response line. And what we would be able to do is call up the tile line map on that same Android tablet and just check and see, is that line already mapped? And yes, it is. So we don't have to spend any more time mapping it again. But if you come across something like a tile blowout or some crop response that's not mapped, you could quickly add that into the system. So we talked about the dry soil lines this time of the year is also, it would be nice if there was one day a year where you could just hire an airplane to fly over everything and you could get it all captured, but it doesn't seem like it's that easy. You'll have a certain neighborhood that is showing up pretty good on one day, and you can drive 10 miles and it's not showing up somewhere else. So maybe that's the lake effect we have over here in eastern Wisconsin. But, and one could, actually drive these lines with an ATV. It would be time consuming, but it's, would do a nice job of logging them into the system. So this is an example of a tile map that we develop. We like to use different color codes based on how was that line determined? Was it apparent from aerial imagery? Was it mapped from crop response? Was it actually logged after an actual installation so we know it's there? Or was it drawn in by the farmer because he knows a line goes from here to there? And I should mention that the other features, the inlets, the vents, the tile blowouts were also on that map. So one of the things we can do then was, once we have this system, we can export those lines, those features, as a KML file, which Google Earth is free to everybody. A lot of farmers have it, both on your phone and your laptop. And just the simple act of emailing a KML file to someone and opening it will launch Google Earth, and it's an easy way to share tile features maps with your clients. And same thing goes with a smartphone. You open that in your smartphone, and now you've got a live GPS map of your tile system that you can use in the field. And we commonly have clients will ask us to email that to them because they're trying to fix something, looking for something, and it's not perfect. It's not going to get you right within 3 feet, but it's a map that's live, and it's going to help. So this system could be used by agronomist, consultant, farmer, custom haulers. And what's really key is that it needs to be a cloud-based system, and that's how most systems are now. It's going to take a little time for farmer scouts and custom workers to input data and to actually learn how to use the system. But once it's up and running, everybody can have access to the same data. And I mentioned it does take a time commitment to, you see something, stop, take a photo of it or log it, get it into the system so everybody can benefit from that. The system we use would work on Android or Apple devices. With this system, drainage system maintenance becomes easier and environmental risk is reduced. And one of the things when you start to talk about tile maps with farmers is that not all farmers welcome the idea of sharing a map that shows the location of their tile lines. But I think as time has gone by and everybody sees these images, it's obvious that really anybody could have access to anybody's tile maps if they wanted to spend the time doing a little research. And in a system, in a cloud system, a consultant could control who has access to each set. So we put things in files, and if a certain client wants their information, we can share it with them, and they don't get, the information for their neighbor's farm. So what the future holds as aerial imagery becomes more sophisticated and affordable, tile feature mapping will improve. Sensor technology such as thermal images on bare soils will likely be useful. I have not personally seen that yet. Hyperspectral imaging has potential. That also I'm very curious about, have not seen that yet. We mentioned the machine learning. That is very likely is going to happen, but those maps are still going to need to be ground truthed. And also UAS systems could potentially be programmed to fly the drainage lines looking for blowouts. That would be my presentation. If anybody has any questions, you could contact me, or not sure, Erica, if we have time now.
If anyone has any questions, you can feel free to type them in the chat. We'll maybe wait a minute or so, see if anyone has any questions for Steve. Okay, I'm not seeing any at this time, but Steve's contact information is shared there on the screen now if you want to take that down. And then I think we'll move on. Our next presentation is going to be from Aaron Wonderland, and he is a part of our Discovery Farms team. He's over in the eastern side of the state, and he collects all of our tile samples. And today he's going to be talking about some preliminary Discovery Farms tile data from our project in 2018 and 2019. So Aaron, I'd invite you to share your screen now.
Nothing yet.
Looks like it's going.
Yeah, my computer's doing a lot of weird stuff. So
I think it's a little bit bogged down because of the participation.
okay.
If you want to click on display settings, so it's in present or not in presenter mode. Yep.
Okay. All right, thank you, Erica. So I will be talking about the preliminary data evaluation from our tile project. It is still preliminary. We don't have all the data. We haven't done a whole lot of statistical analysis on this, but it's showing us some pretty neat stuff, and it'd be interesting to see how statistically correct some of these trends are based on what we're seeing. So, and I think Erica mentioned this. We did get a CIG grant to work on monitoring tile project. And this is in partnership with Minnesota Discovery Farms, and it's a 3-year project. Started sampling in 2018, and it runs through the end of this field year. So there's 3 main goals with this project. First is to understand what nutrients and sediment losses are in these tiles and how the management practices on that field are affecting this. And with that information, we'd be able to pass it on to farmers and consultants, and they can use that to so they'd have more information when making their management decisions. Second goal is we're trying a few different monitoring approaches, and what we're hoping to do is to see if there is an easier, cheaper way to collect the data and have good data, but also it can potentially be a method that farmers and consultants can use so they can monitor and understand their own tiles better. And then the third goal is to understand the link between soil health and tile drainage. So our monitoring design has 3 different styles or levels of monitoring. We have first is intensive sites. We have 8 of those total, where we would do continuous flow monitoring and continuous water sampling. Second level is intermediate sites. We have 20 of those total, where we have continuous flow monitoring and biweekly water sampling. So every 2 weeks, we'll go out and take a grab sample. And our third level is basic sites, and we have 20 sites total for that. And we're doing biweekly flow monitoring and biweekly water sampling. We have 48 sites total, half of them in Wisconsin, half of them in Minnesota. And at the end of the study, we'll have 144 site years of data. In Wisconsin, we have 4 intensive sites. This is your typical USGS monitoring, where you have the monitoring station, you have a refrigerated sampler, you have a bubble system that tells depth of the water, and you have the data logger, which holds the program for sampling and also stores the data. So what the program does is tells the sampler to take a sample, and that's based on time in between samples, but also changes in flow rate. And we access the tiles through an AgriDrain water control structure. We have 9 intermediate sites in Wisconsin, and again, we access the tiles through an AgriDrain. And here we use a level logger probe to measure depth of water. And to take a sample, we come every 2 weeks and use a logger handled dip cup to reach in and grab that. So why am I mentioning depth? We can convert the depth to flow using rating curves for V-notch weir that we had custom made and put into these AgriDrains. So on the left, you can see kind of a close-up of the weir, and then on the right is just a view of water flowing through it. And then Wisconsin also has 11 of the basic sites. This is where we go to the tile outlet. We use a bucket and stopwatch to collect water for a period of time and then calculate flow from that. And then we take samples right from the end, put the bottle into the water stream coming out of the outlet. And again, we do have a soil health component where we're testing several different soil health metrics. And we're looking for a link between soil health and clean water. And Matt kind of talked about that already, so I'm just going to focus on talking about the water quality results from Wisconsin. And this is going to be the biweekly data collected. And keep in mind that this is still preliminary data. So not all tiles flow the same. In this graph, we're looking at the frequency of flow based on our visits. This is 2 years of data, so there were 52 visits. And if you look at those first 3 on the left, we had 100% flow frequency, which means at each of the 52 visits, there was flow. More than half of the flow, or half of the sites had flow more than 80% of the time. We only had 2 sites that had flow less than 50%, and one of those was less than even 25%. And if you compare this to Minnesota, Wisconsin has a much higher flow frequency than Minnesota. When we compare the states, we have Minnesota on the top here, Wisconsin on the bottom. On the left, we have suspended sediment. On the right, we have total phosphorus. You can see that Wisconsin has a lot more variability in the concentrations in the samples. With sediment, the median concentration is slightly higher than Minnesota, but with total phosphorus, Wisconsin median is slightly lower than Minnesota. On the left here, we have in the light purple is dissolved phosphorus. And again, you can see that variability. But in this case, comparing it with Minnesota, the median concentrations are the same. And the reason we're seeing this variability in those first 3 constituents is that Wisconsin has a lot more older clay and concrete tiles that are reaching the end of their lifespan. And it's allowing more soil sediment to get into the tiles. And so we're seeing that you get the phosphorus coming out in either particulate or dissolved. And so we're seeing that variability because there's more, basically there's more getting in. On the right in the green, that's nitrates. And you can see that in both states, there's quite a bit of variability. But in Minnesota, we're seeing an appreciably higher median concentration in the samples. And we believe that the reason for this is that Minnesota has a lot more cash crop systems. They use commercial fertilizers. Those fertilizers convert to the nitrate form much more quickly, if it's not already in that form. Whereas in Wisconsin, manure is oftentimes the nutrient source for these fields. And they break down or convert to nitrate much slower. This is what a typical site would look like in Wisconsin. This is just each of the samples plotted based on the date they were taken. And you can see here that nitrate has more variability than the other 3. Suspended sediment concentrations in tile are typically lower and remain relatively constant. We don't see a lot of big changes, maybe a few little bumps, some small spikes, but they are typically, you don't see very much in the way of sediment. Of course, there are always exceptions. These 3 with the arrow, those are actually warming periods during winter where surface flow is going into the tile through macropores. So you're not getting anything filtered, it's just a direct conduit into the tile lines. The blue arrow, that's actually has a tile inlet right after a heavy rain, and a lot of water flowed towards that inlet and got into the tile that way. The yellow arrow, that's soil disturbance from tillage during a nutrient application that was shortly before heavy rain. And the 2 purple arrows there, they're showing more heavy rains with tile inlets. As far as total phosphorus, concentrations are typically low and consistent, but you can see variation based on surface activity. Looking at the the plots, you can see that they are fairly flat, but there are a few bumps that show up. And again, there are exceptions, and these are pretty much the same reasons as sediment. And if you remember the other graph, you can see that this matches up fairly well. So we have the macropore flow in winter, we have the tile inlet, we have a nutrient application right before heavy rain, and then this is more of that nutrient application showing up as we get more macropore flow, and as soils are thawing and water can go through them more. Dissolved phosphorus, its contribution is most of the total phosphorus. About 80% of the phosphorus in a sample on average is dissolved phosphorus. And it's directly related to total phosphorus. So you can see in the graphs, they are quite a bit the same, with just a little bit of bumps here and there, but not a whole lot of variability. And again, this is a comparison of the total phosphorus with the dissolved phosphorus, and you can see that they look the same for the most part. And these are, again, are the exceptions to that lower, smaller variability that we see in most tiles. But when we have older tile systems, things look a little bit different. And they're noticeable in these first three, sediment and phosphorus. They are still somewhat flat, but there is more variability in the data. Other than the two winter macropore flow bumps, we have several others that show up. And looking into the data, all of these are related to big rain events. So more water is moving through the soil, it's carrying more stuff into the tile. And we don't really see that change when looking at nitrate. Nitrate concentrations are the things that are fairly variable within the tile. And other than those exceptions in the sediment and phosphorus on the top there, again, those are relatively flatter, I guess you could say, less variability. But if you look at the bottom at the nitrate, there is a lot going on down there. Nitrate concentrations are affected by several things, such as the crop that's planted, the nutrient needs, the type of nutrient and timing, and also temperature can have an effect on converting from other forms of nitrogen to nitrate. So first, looking at crop, we can see that there are some differences in the concentrations we're seeing from the different fields or different cropped fields. Alfalfa is a little bit lower than corn, forage, grain, and soybeans. Pasture is the lowest. We have the vegetable, which was green beans, which is also low. But I would like to say that the vegetable field is only 1 year and one site. So we don't have a lot of data points, so we may not be getting the whole picture there yet. Same thing with pasture. We only have one site with 2 years of data. So we may not be seeing the total picture if, you know, compared to if we would have had more sites with pasture and more sites with green bean or vegetables, sort of things. We've also noticed that the previous crop can have an effect on nitrate concentrations. So it's easy to look at the crop that's there, but when you think back about what was there the year before, things look a lot different. When we had alfalfa following alfalfa, it was much had much lower concentrations than alfalfa following corn. And looking at corn after alfalfa, it's also much lower than corn following corn or grain or soybeans, and even the vegetable or green beans. But again, that is only 1 year at one site. So again, that might not be the whole picture for that. But previous crop does show that there is an effect on nitrate concentrations. So nitrate concentrations do vary based on the crop and nutrient applications and temperature, and these cause seasonal variability. And you can see that in this chart. You can't see that so much in the sediment and phosphorus charts. This first section here, that's coming into the project. Those top 3 are either corn or soybeans, coming out of corn or soybeans. Their concentrations are higher than the other ones. Coming out of winter, we're seeing increase in concentrations as the soil's warm. If you look at the taller peaks there with the upside down triangles, that's actually a field that's going into corn, but they had manure application the previous fall. So there's a lot more there, and we're seeing it as the soil's warm. And in this year, 5 of the 7 sites had corn planted, and the other 2 were wheat. Once it gets into fall, then soil's cool. You're not getting you're hardly getting any conversion to nitrates, so concentrations fall. And then in this last year, again, when it warms up, you're seeing an increase in concentrations. The 2 at the 2 higher sites, those are the only 2 years that were corn, and they were the sites that also had manure applied the previous fall. So we can see the manure has an effect in the spring. The concentrations go up as the soil's warm, but we're not seeing any effect really in the fall that it was applied. And that's because when those sites had manure applied, the soils were cooler, so you weren't really getting any of that conversion into the nitrate form as compared to spring when it warms up and you get that conversion going on. One interesting thing we noticed was there's these 2 sites down here at the bottom. They're relatively flat. You don't really see much in the way of seasonal variability. It doesn't matter what the crop is. We're just not seeing much change throughout, the period that we're looking at. We're also seeing that those aren't the only 2 sites that we have this flat appearance. You're looking at that, it almost looks like what we would see with phosphorus or sediment. There's very little variation. And when you look at the sediment and phosphorus at these sites, again, you really have to look closely at the sediment and phosphorus. It takes a lot to see some of those just tiny little bumps that show up. Other than that, the big bumps or big spikes in the sediment where we had macropore flow during a time in winter when it warmed up. So the reason that we're seeing this is because those sites are associated with an outside water source. This graph is showing the intermediate intensive sites that have continuous flow. We don't have the annual flow calculated for basic sites because it's not continuous flow. But if you look at these sites, there's a lot more water moving through these systems. On the left of the green bar, that's 2018. That first 2C, that's almost all of the rainfall of that year at that site. Go to the one labeled 3C, that 34.48, that's actually more than what rain fell that year. And that 5C on the right of that box is less prominent, but it does stand out from those other sites. If you go to that next year to the right, 2019, again, those sites stand out compared to the other sites. We had a lot more rain this year, so we are seeing more flow through the tiles, but again, these sites stood out. And that 2C, again, was most of what fell in that, most of what rain that fell in that area. 3C is a lot more than what rain fell in the area, and the 5C is also almost all of the rain that fell in that region. So we're seeing a large percentage, comparing it to how much rain fell, a large percent of water compared to the other sites. And this outside water source would be things like, upwelling of groundwater or perched aquifers with, lateral movement through the soil. So again, with these sites, we're seeing a dilution effect from these outside sources, but we're not actually seeing a dilution from flow volume. The purple triangles are the actual sites that have the outside sources, and most of those are at the bottom of that chart. And whereas the green, which is just what we're seeing is the infield drainage, is more towards the top. If you look at flow volume across the bottom, you see that you're not really seeing an increase or decrease in concentrations in those sites as the flow increases. So going forward, we're going to continue monitoring until the end of September this year. We'll have our full 3 years of data, and then we'll get into more of our data analysis and evaluation, including doing some more statistics. We'll look at effects and what's causing things during events. We'll look at the effects of management and any other investigations that kind of pop out when we're going through the data and look interesting, see what's going on. USGS is also doing some modeling. They're going to look at the nutrient and sediment loading from the tiles. They're also going to compare the monitoring styles to see how closely they relate. They haven't gotten too far into this yet. I think they're just still trying to figure out how to put the model together and making sure that it's accurate and correct. But from what I've heard so far, they said it looks like it looks good, I guess is what they're saying. So, but we still have more data to add and more of the regions to look at. And then we're also going to continue outreach. We're going to talk to the participating farmers, go over what we're seeing on their systems. We're going to do webinars, conferences, meetings, things like that. We're also going to do articles and other publications. And all these are ways to getting the information out that we're learning so it can be used by the people that can use it and not just sit in some database somewhere and not get anything out of it. So with that, if you have any questions, I do have my contact information here if you think of something later.
We do have a couple questions in the chat for you, Aaron.
Okay.
So the first one is, how often are the thermal image maps made available? And annually, and are they regional or statewide?
The thermal imaging maps? I think that might be a Steve.
Yeah, that's a Steve question.
Oh,
Yeah.
you're right.
So that would be something that's not widely available, that you'd have to hire an aerial imagery company to fly that. I know it's available to hire, but it's just not out there to, grab off the internet.
I think this one's more of a comment. It says, looks like the pattern for nitrate concentration seems to be a mirror of P. When P is high, nitrate is low.
Yes, we also noticed that. We still need to look into that a little more to see why that is. Some of those, that we looked at, it looks like when nitrates are lower, we're getting surface water through macropore flow during the warming in winter. So maybe we're getting kind of a dilution effect in the sense of getting more water in the tile. Because we don't normally see a lot of nitrates lost in surface runoff. I mean, we do get losses, but not as much as we would get when we're looking at tile.
And then is there a difference of the average depth of old tiles versus newer lines?
I believe all of these tiles are in at the same depth at 4 feet. I don't remember seeing any that were any deeper or shallower.
Okay. The next question, is there any attempt to partition between the water that has reached the tiles through preferential flow and the base flow or matrix flow?
Is this referring more to that outside source?
Actually, if you don't mind, the couple of questions on both preferential flow as well as nitrate, if we can kind of hold those until after my talk, because I will be covering some of that will hopefully shed a little bit of light on those before we start having that discussion. That'd be great.
Okay.
Thanks, Eric. Anyone has any more questions for Aaron? You can type those out. Seeing none, we'll thank Aaron. You can go ahead and stop sharing your screen. His contact info is there. And we will move on to our last presentation, which will be given by Eric Cooley. He's the co-director for the Discovery Farms program, and he's going to be talking about tile data from some previous projects and what we know.
All right. Well, good afternoon, everyone. Are you able to see now my screen correctly?
Yep, you can put it in slide mode.
And now are we seeing it right, or do I have to switch screens again?
Looks good.
All right, great. Well, thank you, Erica. And so what I really wanted to try to do today is talk a little bit about what we've seen in the past, and some of the main lessons that we've learned, and how that really is translating to some of the things that we're seeing in some of this new information that we're gathering through the CIG tile project. I would really like to recognize Tim Raditz from the Minnesota Discovery Farms program that has really helped to put a lot of the graphics together for the presentation that I'm going to be providing today. So Aaron did, I think, a really great job of talking about kind of our current tile project that we've got going in the past 2 years. I know he talked about the 48 sites that are being done in Wisconsin and Minnesota, but Minnesota actually has another 5 sites that they are monitoring that are not part of that project for tile. And this really is building upon the information that I'm going to be talking about, which is the monitoring that both states have done from 2004 through 2017. And you can kind of see kind of comparatively how we're really ramping up in these last 2 years, the information that we're learning from tile. I think, tile is one of those things that we're starting to understand a few of the concepts, but we really do have quite a bit more to learn. The great thing about the databases that we're sharing between both the Wisconsin and Minnesota Discovery Farms programs is that most, if not almost all of our systems, especially our historical systems, have been manured and with respect to tile drainage, whereas Minnesota, conversely, has very few manured systems where most of those are cash grain operations or some other vegetable or specialty crops. So it really is nice to have a mix of the two data sets to kind of compare and contrast. And you'll definitely see some differences, as Aaron pointed out, and we can talk a little bit more about those as we go along here. So again, looking at this combined data set from basically 2004 through 2017 in both states, when we look at the surface runoff coming from tile drain landscapes as compared to tile flow, we can see that from a total water amount coming off of those two different venues, we can see that the tile flow is about 1.5 times that typically as compared to a surface water flow. So we're only seeing about a little over 1 to 4 inches that we are seeing from the surface, whereas we're seeing about 2 to 7 inches typically through those tile systems. One of the big things that we do see, though, that is a big difference between the two is that for surface water flow, we're only averaging about 8 actual days where that surface water is flowing, where we're seeing at least cumulatively about half of the days, about half of the 365 days a year, we're seeing tile flow occurring in those systems. From a surface water perspective, we're pretty evenly split with between frozen and non-frozen soil conditions with the runoff occurring. Whereas tile flow, again, the two states combined, we're seeing the vast majority coming through non-frozen soil conditions as compared to frozen. But we're really going to see a differentiation between the two states where in Wisconsin, we are seeing a lot more flow throughout the wintertime as compared to Minnesota. So again, looking at this graphically, surface water data, which is this first line that's represented there, is really quite similar between the two states. We can see that there's a first peak where we have snow melt in March, where we really see the majority of the runoff occurring on an annual basis. And then oftentimes we're seeing this secondary peak with those late spring rains right around the time of planting, where in May and June we have this other peak that occurs in surface water runoff. Now, when we look at Minnesota tile flow, in fact, when we went over to try to first get into tile drainage monitoring, they said, it's a piece of cake. It starts running a little bit in spring, may run for a couple of months, and then you're done for the rest of the year. Well, their data that they've collected really has shown that, where you really don't see much tile flow until you start getting that snow melt in March. It picks up. The peak really coincides with that secondary surface water runoff in May and June. And then it basically trickles down throughout the growing season and shuts off until the next snow melt the next year. Wisconsin, conversely, we really see our peak during that snow melt in the March time period with snow melt, and it really remains high through June as it tapers off. We get it dipping again, like Minnesota, as you get into the growing season. But unlike Minnesota, we see it start to pick up in late fall in November and December, and then again ramp up in January, where we get those late fall rains and some of those preliminary snow melt events that occur on those soils. So very different flow patterns between Wisconsin and Minnesota, and that will translate to some of the differences that we are seeing in some of that data. Go ahead. For those of you that have been to any of my tile talks, you've seen this slide, actually the next couple slides, because macropores, especially in Wisconsin, we really believe are driving a lot of the water as well as nutrients and soil that we're seeing in some of our tile systems. These macropores can be a variety of these different preferential flow paths through the soils. One of the most common one is earthworms. If you're an earthworm living in a tile drained field, you're going to build your home right over that tile system, because, you know, much like the picture that Steve showed, worms do not like to have saturated conditions for long periods of time. So we have seen when they've done worm assessments, there is a much higher density of earthworm burrows directly over a tile line as compared to the areas in between. Old root channels that decay and leave a void in the soil can also be a preferential flow path. Shrinkage cracks, especially in our soils in northeast Wisconsin, which are heavy clay. You know, we've seen these surface cracks that develop when those soils dry out that can transport material down 17 feet or deeper in some test pits that we've dug, where we've seen litter from the soil surface getting deposited 17 feet down in the soil through these shrinkage cracks in the soil, as well as the natural structural porosity. And as that picture on the right shows, with some dye that was applied to some soil, you know, the preferential flow through that soil can take a variety of these macropores and use different combinations of these as they're going down to those different tile systems. But again, the macropores and the preferential flow can really be a strong driver in some of the data that we're seeing in our tile systems. Again, one of my very favorite graphs that I'm sure many of you have seen, but it was a real eye-opener for us. And again, we're seeing this in our more recent data that Aaron just talked about, where this is one of our first years of tile drainage monitoring, going from 2004 into 2005, a very cold winter where we did not have a deep snowpack. So we had over 2 feet of frost in the soil as we're getting into this late March time period. We've got the air temperature there in red with the dashed line showing the freezing mark with your legend on the right. And then we've got the tile flow in brown, as well as surface water flow in green with your legend on the left-hand side there. And we can see that as we're starting to get into spring, and we're getting this above freezing temperatures during the day and below freezing temperatures at night, we are seeing tile flow 24 to 48 hours before we're finally getting enough melt that it is running off from the surface during the daytime. Again, this is soil that has 2 feet of frost or more in it. And we are seeing water that is being transported from the surface into the tile system through those macropores. And in fact, when we did the water quality analysis between the surface and the tile water in this basin, they were almost identical with the concentrations of the constituents that we monitored. So we know that the water that was being transported during this time period was going through those macropores and not through that soil matrix, which really again drove the constituent concentrations in those soil or in the tile systems. We've also seen, because of the really good macropore development, especially in long-term no-till systems, that they are extremely good at transferring water into these tile systems effectively. What we've got here, again, we've got our surface runoff in a basin in green, we've got our tile in brown, and we've got rainfall below. This other little dashed line that I've put on this graph is showing when those tiles are flowing at capacity. So as we get into the second of this month, you know, tiles are kind of draining. We've got 0 here for surface water runoff. All of a sudden we get a rain event and we see a very rapid response in the tile. And until that tile is flowing at capacity near, we don't see surface water being initiated. And we can continuously throughout this time period see that we aren't having surface water flow unless those tiles are basically maxed out. So we can see this first event, tiles are maxed out, we get surface water flow, they start to fall below that maximum capacity, and that surface water shuts off. Again, they start to trickle down, we get another rain event, bang, they pop up, and again, until they're flowing at capacity, we see that surface water expression, where once those tiles can't handle anymore, that's finally when we see the surface water running off. In fact, one of the things that we've noticed, especially in some of these very intense rainstorms where they had a large initial intensity, we saw an effect in that tile system between 10 to 15 minutes from the time that that first raindrop fell on the soil until we saw a response in the tile system. So again, this macropore flow to these tile systems can really rapidly transfer water from the surface down into the tiles, oftentimes with little to no filtration. So when we're looking at soil loss, again, going through many of the same things that Aaron just did, again, only on a kind of a more historical data set, we really do not see much soil loss that is occurring through those tile systems as compared to surface water flow. We can see that dramatic difference with surface water on the left, tile flow on the right, and they are many magnitudes larger in the surface than in the tile. Interestingly, when we look at all of our tile sites and graphing them from the lowest soil loss on the left-hand side to the highest soil loss on the right-hand side, we kind of see this natural break that falls where we really are just not seeing much of anything for soil loss through those tile systems. And then finally, we start to see some additional on the right-hand side. And if we look, that those first half of tiles are basically your newer corrugated plastic tile that have that really defined opening or slits that are in those tiles that really control soil movement into those tile systems. If we look further on the right, we've got a number of reasons why we begin to start seeing more soil loss to those tiles. We've got some older plastic tile at the first site there where it was starting to deteriorate. And then we start getting into the clay and concrete tiles that again don't have that well-defined gap that water enters those systems, which does allow some soil to go in. But then as we get into these end ones where we're seeing quite a bit more soil in those tile systems, that's where either those clay or those concrete tiles are really hitting the end of their life expectancy, and we're seeing the formation of tile blowouts and the introduction of much more soil into those tile systems from those blowouts. What we tried to do is provide a graphic along the top here of how a blowout forms based on some information that we've heard from tile installers, where basically you get a little weak point that forms in a tile, whether it's a junction or just a breakdown of the tile material. What it does is it will pressurize, in that second photo, the soil right above it. And as you get this cyclic pressurizing and then falling of water levels in the soil, it brings soil particles in, which then creates a void that you can see in that fourth diagram, which will eventually collapse at the surface. And as Steve nicely pointed out, as we get into snow melt through the spring of the year until those crops get established, is a great time for trying to identify these out in the field. Especially after snow melt, we can see on this left-hand picture water actually bubbling up through a tile blowout that we had found. And the other thing that we can see too, especially when we're having that snow melt runoff, is that you can actually hear some of these blowouts as there's a vacuum almost on some of these, that you can hear it sucking water from the surface down into the tile systems. So again, this early spring period is a great time of year, especially when you're having a little bit of surface runoff to try to identify some of the tile blowouts in some of these older tile systems. So again, looking at some of the phosphorus loss between surface runoff on the left and tile flow on the right. Again, much like soil loss, we're seeing much more loss from the surface as compared to tile flow. But interestingly, we see some differences between the speciation of that, where again, we're split about half between particulate and dissolved phosphorus from the surface water systems, whereas in tile flow, we believe that because of preferential flow, that is predominating in a lot of these soils that are tile drained, we are seeing a much higher fractionation of dissolved phosphorus in those soils as compared to particulate phosphorus. And this is really, this is really validated, you know, when we're looking at soil test phosphorus on the x-axis here and the dissolved phosphorus concentration on the y-axis. You know, we already know that this is pretty well established on the darker purple line from the surface, where, you know, we see a very strong correlation with that soil test phosphorus to dissolved phosphorus concentrations that are running off the soil surface. But again, because of preferential flow, we are seeing some similar trends in the tile systems as well as the surface. So again, this gives some additional evidence to the importance of preferential flow to these different tile systems that we are monitoring. And then finally, getting into nitrogen, you know, this is where, you know, we really start seeing a departure from the surface water driving annual losses, where, again, looking at surface on the left and tile flow on the right, we're seeing quite a bit higher total losses through the tile systems as compared to the surface. From a surface water perspective, most of our, about 3/4 of our loss is in the organic and ammonium forms as compared to nitrate, whereas, you know, it is predominantly nitrate in the tile systems as compared to the organic and ammonium fractions. Now, this is one of the graphs that that Tim just created that I just love to show, and really builds upon, again, some of the information that Aaron just talked about, where when we're looking at the annual flow on the x-axis versus the annual nitrate loss on the y-axis, you know, we do see that flow is really the main driver for loss. When we're talking about annual losses from tile systems, flow is the main driver that we're seeing. So on years that we have very low flow, we typically see low loss, and when we have high flow, we see high loss. What we are not seeing a very strong correlation is the dilution effect of water that is coming through the soil profile to these tile systems. So especially when we're looking at, you know, some of these lower flow values, we've got some very high concentrations as well as very low concentrations at similar flow values. Where that really starts to kind of miss is, as Aaron talked about, when we're having water that is coming not through the soil profile, but, you know, flowing laterally from other areas and intercepting groundwater where we have very high annual flow, there is a dilution effect because that water is, again, not coming through the soil profile, and we are seeing more flat-lined and relatively lower nitrate values in those tile systems. Oops. On average, we're looking at about 4 pounds per acre of nitrate for every inch of drainage. Again, looking at that, that fit line that we have on the graph. But one of the things that we can control, again, breaking these up into the different quadrants, is the concentrations. And this is where I really think that, you know, not only our historical data, but our new data will really help to provide better information on how we can better manage our soil nitrate concentrations to try to mitigate loss. Because just having, you know, a similar annual runoff value of 6 inches, you know, we can see that our total annual losses would be tripled by having higher nitrate concentrations in that soil, which is one of the factors that we can really control. And this is where I think that, you know, we're hoping to provide more information from the data that we are collecting with soil nitrate levels. This is a great publication that Purdue put out, really talking about the values of nitrate in tile. And, you know, we're looking here at the 10 to 20 parts per million range, or milligram per liter, is really what we would expect to see where we're applying N rate to row crops that are meeting those needs. But when we start to get over the 20 part per million range or milligrams per liter, we're really seeing where we're either not timing that correctly or that we're applying in excess of what that crop really needs. The one last example that I wanted to just show quick was, this is some Minnesota data where we've got corn grown 3 years. There was a year of fallow in 14 and then followed by 2 years of alfalfa. The first 2 years, there were spring applications of manure to basically meet the nitrogen requirement of that crop. And we can see that with those spring applications, we're averaging between that 20 to 30 part per million range. But then coming out of 2012, again, going to back-to-back corn, there was a fall application that was made very early in the fall where it was very warm soil conditions. And again, like Aaron talked about, we saw the conversion of that nitrogen from organic and ammonium forms into nitrate. And as we got into the springtime flow in that tile system, we saw those soil nitrate concentrations being transferred to the tile and really remaining high prior to that crop growing in the field. So again, anything that we can do when you have to make those early fall manure applications, such as trying to wait till those soils cool or providing a cover crop to really try to maintain that nitrogen into the following year and prevent that conversion, will really not only mitigate some of the losses, but retain that nitrogen for the following year's crop growth. So with that, I just wanted to let people know that we do have quite a bit of different publications and information available on both our website as well as an FYI extension website. I know John Panuska has been great at trying to update that site, and we'll be definitely putting more information onto that site. Erica, if you don't mind, could you throw up that first survey? Because one of the things that we're trying to do is that we're trying to provide information that really meets the needs of farmers and their consultants. So, for example, we just put together the Guide for Tile Drainage Compliance in Wisconsin, and that was really, it's not based on any data that Discovery Farms has collected, but it was really seen as a need, and it was pointed out to us by the industry. So we're really looking for your input on not only what other information you would like to see, but then we're hoping to put a field day together, hopefully yet this year, depending on how things go with the epidemic, but if not, next year, and would really like to try to find out some of the things that we should focus on. So if you don't mind filling out that survey, that'll really help to guide the efforts of publications, research, and things that we'll be trying to put together in the upcoming field day. So with that, I'd be happy to take any questions. I think Aaron and I, Aaron, if you don't mind coming back off of mute, we can try to tag team some of the questions that were talked about earlier.
There is one more question in the chat. To what extent did climate change affect the trend of tile flow peak variation?
Great question. One of the things that's a really great question, because we are seeing more intense rain events rather than the really long drawn out rains that we typically have seen historically. So one of the things that I would say is that especially when we have these higher clay soils that have that need to kind of swell back up for those cracks to stop transmitting water quickly to the tile, that has resulted in more of that instantaneous flow from the surface to the tile rather than through that soil matrix. So I do think that is one of the things that we are seeing with some of the more intense rainstorms that are now more commonplace than in the past. but to answer your question directly, I think someone else has a better idea. I don't have much other than that.
Back to the questions that were asked previously.
Sure.
There was one, is there any attempt to partition between the water that has reached the tiles through preferential flow and the base flow or matrix flow?
Yes, great. Thank you for bringing that one back. That is definitely something that I know Aaron and I are going to be working with Matt Ruark and others to try to, maybe John Panuska, to really try to do the calculations. I've actually been reading up on a number of different papers, and really trying to parse those two different pieces out is very challenging with this, not only the calculations, but some of the basic dynamics in the soil. We are going to try to quantify that, but again, we're hoping to make progress in that in the next year. So,
And then you might have talked this a little bit, but any thought that higher nitrate in Minnesota tile lines is related to flow? Generally much lower overall tile flow in Minnesota than Wisconsin. Thank you.
So again, one of the things that I will say that goes back to some of Matt's talk on soil health and how we're trying to understand these different dynamics is I truly believe that when we're looking at manured versus non-manured systems in Minnesota, again, a generalization, we are seeing relatively higher soil nitrate concentrations in cash grain operations as compared to manured systems, whether that's effect of the type of the fertilizer type, or whether that's a function of soils or flow patterns or a number of other things. We're still trying to, I guess, hone down on the reasoning for that. But also in some of the tile monitoring that's been done under cash grain systems in Wisconsin, again, we're seeing some similar trends that the nitrate values in that are more related to the cropping system and crop being grown rather than the actual flow. I'm sure that there still are some flow effects, but I think that they are are much less dependent than the actual fertilizers and crop being grown.
Good. I'm sharing the poll results now. Unfortunately, the question 2 was marked only as a single answer.
Okay.
So we'll share that in the survey that goes out this afternoon also. So people can answer that question fully. We will be sending out a survey post this webinar. So watch your email for that. And if you attended, please fill that out. One reminder before we answer a few more questions, if you are a CCA hoping to earn CEUs, to make sure you had typed your name into the chat box. Eric, John was wondering if you could talk about roots and tiles.
Sure. And before I do that, I want to again thank all the people as people start to tailor off here. I want to make sure to thank all of our speakers today. Matt, Stephen, Aaron, I greatly appreciate the information you provided. Even after this, I want to make sure that anyone on that listened to today's webinar or is listening to this at a later date, make sure to contact any of us for more information, because we're desperately trying through any types of media to try to get this information into the hands of people that can use it. So if you ever have ideas of things that we can do better, publications that you're looking for, information, if I don't have it, I will try to get you the resources that do. So, okay, getting to John's question. Tiles or the roots in tiles. Yeah, that is something I know as Aaron Wonderland and I went through our meetings with our farmers in Wisconsin this year, that topic got brought up more than I have heard in the past. And I do think, especially, I'd be interested to hear Matt Ruark's comments as well. But as we're looking at some of these cocktail mixes, especially with brassicas, in those, we are starting to see some challenges with these cover crop cocktails of roots getting down into tile systems, as well as willows and other things near drainage ditches. So I do know that that is a challenge that is that can really hamper tile system function, and really need to be kind of aware of that. And we do have some information in our publications to address that. And hopefully, if you need more resources on that, please contact me and I will try to point you in the direction. And John, if there's anything you want to add, please do.
Eric, I remember when we talked about this at the tile training, you had mentioned something about that during the off flow season that those roots tend to dry out and can often be washed out of the tiles. So just a little anecdote there to throw at it too, in terms of, it's not like these things are going to be there forever unless they're willow roots. A lot of the grasses might just dry up and be washed out.
One thing I noticed a couple of my field visits, we did have root mats caught up in our AgriDrains that I had to clear. So yeah, the roots do come out.
And I do see another question too about iron ochre, and Aaron and John, if you want to talk a little bit more. I know that at least at one of the tile sites where we've got one of the controlled drainage, that we have seen iron ochre issues in the past, that we actually tried to flush that system this year. And again, Aaron or John, if you want to talk any more on any developments with iron ochre.
Yeah, I can throw in a few words. I think most people understand iron ochre is really becomes a problem when you have high iron concentrations in your groundwater or in your drain water, and that iron ochre is a kind of a gelatinous, filamentous, organic goo, so to speak, that can get into a tile and really cause some problems. I don't know, Eric, if you had any luck with flushing, but about the only real environmentally acceptable way of clearing that stuff out is with pressure jetting, and try to jet it out and get it out of the system. We do have several publications on the Drainage FYI site about iron ochre, and I'm continuing to watch for others, and I'll post them as I find them. But check out the FYI site. There's some information there.
Great, thanks, John.
There's a question for you, Eric. Did you see the same amount of preferential flow in Minnesota as in Wisconsin?
That's a great question, and one that I'm going to be connecting more with Tim on. I do know just from our data that, and just knowing the soils and the differences, that we typically have seen the effect of that more, especially during those wintertime months, than we have in Minnesota, where they're just not seeing that preferential flow effect in the winter, or during the winter time and coming into snow melt in the spring. But as far as more of the recent data, if they're seeing different soils or different landscapes where they are finding more preferential flow or different longer term no-till systems, that is something that I do plan to follow up on Tim with. But kind of in general, I would say that that we're seeing, for whatever reason, more preferential flow in Wisconsin as compared to the Minnesota tiles that we've monitored.
Looks like we have one more question. Can the tile drainage N data be related to NUE? And do you have the data needed to evaluate this at some of the sites?
Ooh, great question.
I can take this real quick a little bit. I talked with Abby, who is our NUE coordinator. She is looking for the data on our tile fields so that she can run NUE numbers. So we haven't really done anything with that yet. I don't know if anybody else has, but that would be something that would be coming out at some point.
Great, thanks. I'm not seeing any more questions, so I'm going to go ahead and stop the recording, but